Okay, so just a big disclaimer. So all of the material is written by Derek Chen, and he's unfortunately sick today. So I'll just be covering for him. Like I'll literally just read off the script that he wrote for me. So yeah, all the credits goes to Derek. Right. So so before we begin, I want to ask you all questions. Um, so when I put together the words of RNA and the field of medicine, what are some of your first thoughts on these topics? So you're probably thinking something about this. Um, well, today I'm going to talk about the potential of RNA not only for vaccines but also about their uses as therapeutics as well. So to show you the progress already being made in the field, here is a list of all RNA drug candidates in clinical trials or is going to be released in the near future. As you can see, this is quite a long list, but before you get into the details of the different kinds of RNA therapeutics, let's go back and review some of the basics of RNA. So let's talk about what's RNA and have a little review of the IASM block. RNA is a polymer of nucleotide joined by phosphodiester bonds, and it's usually single-stranded. And it's compared with DNA, uses ribose in its backbone and the nitrogenous bases uracil instead of thymine. So again, for review, a nucleotide consists of three components. You have a sugar, a phosphate, and a nitrogenous base. So for sugar, the sugar we have, the ribose, in RNA is that, sorry, no, okay, right. So ribose is a sugar that RNA use instead of deoxyribose that's used in DNA. And the nitrogenous bases, we have adenine, guanine, cytosine, and the fourth could either be a thymine. Then the fourth one is thymine for DNA and uracil for RNA. So adenine bonds with thymine or uracil, and cytosine and guanine bond together. Adenine and guanines are double rings, and purines or thymine and cytosines are single ring and pyrimidine. So now back to RNA. In our body, we have 7 naturally occurring types of RNA. We have mRNA, which we all know carries information for protein synthesis from the nucleus to the ribosomes in the cytoplasm for the actual production of the protein. And rRNA is what makes up 60% of the mass of ribosomes and then play a major part in the function of ribosomes, such as binding to mRNA and also recruitments to tRNA and catalyzing the peptide bond during synthesis. And then we have tRNA. So tRNA helps with protein synthesis directly. They are non-coding and carry amino acid directly to the ribosome. They are 18 nucleotides long and take a clover leaf shape. Now we have snRNA or small nuclear RNA. So these are RNA molecules of roughly 150 nucleotides long, and they stay inside the nucleus and play a role in helping the synthesis of other RNA molecules. So one such role include playing a role in spliceosome and transforming pre-messengers RNA to messenger RNA. So small nucleolar RNA or snRNA, they are RNA molecules of roughly 60 to 300 nucleotides in size, and then play a role in the synthesis of RNA by cutting down the size of the precursor, rRNA. So second to last, we have the microRNA or miRNA. They are 22 nucleotides in size, and their main role is in RNA silencing, meaning part of the and this part of the control of gene expression. They work by binding to a corresponding mRNA, and as a result, in the repression of translation of that piece of mRNA. Okay, so last of all, we have long non-coding RNA. They are quite a heterogeneous group of RNA that are roughly 200 nucleotides in size. The full function is not fully understood yet, but at the moment, research points to them playing a role in regulating gene expression. Okay, and then we're going to talk about regulations, gene regulation. So it's very important that we have a way for us to regulate things and the amount of genes product that we have. Control of gene expression can happen in a number of ways. This includes control of transcriptions, control of the processing, control of the translation, and control of the protein activity. So first, for the control of transcriptions, we have epigenetics in play. So epigenetics would be things like methylation, etc. And these epigenetic mechanisms will determine which gene are expressed. There's also enhancer regions on the genome as shown in the picture. The enhancer can be bound by an activator protein, which itself can be bound to a ligand, for example, a steroid hormone. And then when there is a binding of the activator protein to the enhancer, a loop will form between the enhancer and the promoter of the gene, and the activator protein is brought into these close proximity to the transcription factor complex at the promoter, so it can influence the transcription. For control of processing, we have influences at the spliceosome. Here, a single mRNA can eventually, at the end, lead to different protein because different exons are exported. As previously mentioned, snRNA play a role in the spliceosome when they are bound to snRNA molecules. So an example of this would be calcitonin, where different splicing can lead to calcitonin being produced or calcitonin-like protein being produced instead. So thirdly, for control of the translation, we have mRNA editing. So in this case, it literally means that the mRNA bases are changed in the cytosol prior to translation. So an example of this is the protein synthesis of ApoB, ApoB100 and ApoB48. ApoB100 is a 4,536 subunit protein when the mRNA is expressed in the liver. But if the same mRNA is expressed in the intestine, there is an intestinal deaminase that converts a cytosine into a uracil, and this leads to a stop codon and a truncated ApoB48 protein that is only 2,152 amino acids long. So lastly, for the control of the protein activity, I guess this is familiar to all of us, and as a result, won't go into details. Example of this is like when cyclic AMP binds to protein kinase A and makes it active. Okay. Right. So why this all matters? Okay. So this is a that was overly simplified statements a lot of the times many diseases occur because something went strange in pathways within our cells. This leads to either too much of a protein being made, or too little of protein being made, or defect in the protein being made. As you can see on this diagram to the right, using the examples of metabolic diseases, RNA therapeutics can be used to modify metabolic pathways. It can be used to modify metabolic pathway in different ways to treat metabolic diseases. It can be a single pathway, or it can be a network of different pathways. While on the left is an example of using mRNA therapeutics to treat disease. The main goal is to increase the amount of protein products. With our current techniques, we use small molecules to correct for these mistakes, to help correct, and we can always look for more small molecules. But there are limitations to that. I will talk about that later. Now, let me talk about the different modalities of RNA therapeutics and how they can be used to address what I just said. And the first example we can talk about is antisense oligonucleotide, or ASO. I think it means oligonucleotide. So yeah, antisense oligonucleotides. ASO are small single-stranded DNA that work within the nucleus during spliceosome process. During the spliceosome process we mentioned earlier. It also splicing, or within cytoplasm, it activates RNase H1. An RNase is an enzyme that breaks down RNA molecules, and as a result, it usually results in less protein productions. So example of this drug would be a drug called nusinersen, which is used to treat SMA. Okay. And the next example we can talk about is siRNA. They are usually double-stranded RNA that are 21 to 22 nucleotide long, and they work to decrease proteins products by a RISC-mediated mRNA cleavage mechanism. siRNA helps argon-UA2 protein to bind to the RNA-induced silencing complex once they enter the cell. Then the RISC binds to the corresponding mRNA as directed by the siRNA and HGO to mediate the cleavage. An example of this drug is patisiran, used to treat a condition called polyneuropathy. Polyneuropathy. And then we have the final example, the one we are all familiar with. So these are ssRNA that's limited by our delivery method to anywhere in size from 340 to 2,300 kDa. They work by ribosomes binding to them to make new protein products, thus increasing the amount of protein products to treat a disease. So all of this is quite new. So in clinical trials, an example would be BNT131, which is used to treat advanced melanoma. It is where a cytokine-inducing mRNA is implanted into a tumor. So I will now talk about the advantages of mRNA therapeutics. Well, first of all, if you want to increase a gene product, using something like mRNA therapeutic is much safer than gene therapy, even if you have to redose as there is no genome integration. Then there is the idea that there are only so much we can drug right now. It's very hard for us to use small molecules to directly drug the majority of the genome, as they don't code for protein, um, and stay as non-coding RNA. But with RNA tools, such as ASO and siRNA, we can directly target these non-coding RNA. So this is especially true now that we have the actual complete picture. Okay, and then so delivery. Delivery is one of our major problems with RNA therapeutics right now. RNA is fragile, and one of the best way currently you can use to protect them for delivery is to integrate them into lipid nanoparticle. Since RNA and its backbone is anionic in nature, when we use this particle LNP that contain ionizable cationic lipids, they can easily stick to the anionic backbone of the RNA and protect it from disintegration in the blood. But current LNP are highly immunogenic. To improve on current LNP and to prevent its immunogenicity, we can try to use biomimetic lipids or lipids that reflect our natural cell membranes as part of our LNP formulation. So alternatively, we can move away from LNP and use cationic inert polymers such as a substance called PBAE to deliver RNA instead of the LNP we just mentioned. So lastly, we can also use our own cell-derived exosomes for delivery. So at the same time, a disadvantage is also because of its therapeutic effects are temporary, and as a result, redosing is required. To begin with, okay, so to begin, in order to prolong the therapeutic dose of any RNA therapeutics, there is the idea of using circular RNA. A lot of the time, we have to constantly redose any RNA therapeutics. This is because in our cell, there are enzymes called RNases, that are quickly they'll quickly disintegrate the mRNA in our cells. And the idea of circular RNA is that it lacks the free end for those RNases to latch onto it, and as a result, these circular RNA will last longer in our cell, and so that more therapeutic proteins can be made. So the enhanced stability is also valued not only in therapeutics, but also in vaccine creation. So, for example, a team recently in the preprint published a second generation COVID vaccine based on circular RNA. So the minus 80-degree freezers that we use for the Pfizer vaccine right now is no longer needed. So right now our current generations of LNP have high affinity to ApoE. And as a result, we'll always travel to where so as a result, they will always travel to the liver when they're injected intravenously. So one of the more exciting breakthrough would be to use what's called Yi-tow hose in our designs. In simpler terms, Yi-tow hose act as sensor inside our cell that can detect which cell type we are in, simply by binding of specific mRNA molecules to it. So when we place it in front of a RNA molecules, we want to make it so that it allows for the fine control in manufacturing of our desired protein. So in detail, this paper describes e-tohos. I think you guys can see the paper, how it's written, e-tohos. So so this paper described e-toho as a modular riboregulators that work in eukaryotic cells. So e-toho stands for eukaryotic Toho switches. So e-toho is an untranslated region on the mRNA whose function is to inhibit translation of the mRNA by preventing it from binding to ribosomes. So only when a specific tRNA, which is trigger RNA, sequence annealed to the Toho switch, does the inhibitory loop get disrupted, and translation can then proceed. This means that RNA therapeutics could be supercharged, as it would allow for the restriction of a specific type of treatment to a specific type of cell, even with systematic dosing. In the investigation, in the investigators' further works, the future may even allow for these e-tohos to act as logic gates, so we can precisely compute the state of a cell and release therapeutics at an even precise granular level. So that is the end of the presentation made by Rick and presented to you by me. Thank you all.